Parasitology, in the parasitology, gastrointestinal protozoans are the major organism in this Entamoeba histolytica, Entamoeba coli, Naegleria species, Giardia lambia. First, we will start from the stool routine examination because the stool is the major test in parasitology. In the stool routine examination, if the stool is a liquid, then within the 30 minutes it is recommended that you have to process it for the trophozoid, helminthes, eggs and larva. If the stool is semi-solid, then 1 hour. Within 1 hour, you have to examine the trophozoid, cyst and helminthes eggs under the light microscope. And if it is form stool, then there will be no trophozoite form, so you can take 24 hours to check for the cyst and helminthes eggs or larvas. Further, OVA and parasite examination, if number of specimen must be 1 or 2, 3. Interfering substances in this test are antibiotic, antiseptics, bariums, laxatives, mineral oil, soaps, water and urine. For the preservation of a stool, we can use the polyvinyl alcohol, 5 to 10% formalin, and FIM, sodium citrate, acetic acid, formalin, SAF, and form stool may be refrigerated for 1 to 2 days. Direct smear saline on a fresh liquid stool to observe the motility of trophozoites. Direct smear iodine 1% D Antony or Logal's iodines to observe the nuclei of the cyst. Trophozoite will be killed in iodine stain. Concentration to concentrate the parasite and separate from the fecal debris. And sedimentation method is a formalin ethyl acetate, which recover the eggs and larva in cyst form. Flotation method, in which the 33% zinc sulfate and the specific gravity is 1.18. Parasite float to the top and may miss operculated eggs and unfertilized ascaris. The permanent stain slides can be is very useful in the diagnosis of protozoa, and for the iron hematoxylin and trichome stain are used widely to preserve the slides. Entamoeba histolytica is amoeba, and it's from the Sarcomastigophora. Its trophozoites are motile through the pseudophodia, and it multiply through binary fission. And this organism have round, fine, even periphery chromatid, small delegate central karyosome. The trophozoite form is 10 to 16 micrometer, average is 20 micrometer, 1 nucleus, delegated finely granular cytoplasm, and ingested RBCs seldom contain the bacteria, single long finger like pseudopod, directional motility. If we see the trophozoite picture, there is ingested RBCs and cytoplasm and nucleus. Nucleus have karyosomes. In the cyst form, which is 10 to 20 micrometer, have 2 to 4 nuclei. And 10% of the cyst have cigar-shaped chromatid bodies, may have the glycogen vacuole. It's cause the acute amoebic dysentery, can cause the extra-intestinal amoebiasis. Only amoeba that ingest the RBCs. Enzyme immunoassays for antigen detection and molecular testing are available. In intestinal amoebas, there are some other amoebas that are non-pathogenic. Number 1 is Entamoeba hartmanni, Entamoeba coli, Endolymix nana, Iodoamoeba busheli. These are non-pathogenic amoebas, and they have very resemblance to the pathogenic strain, that is Entamoeba histolytica. Entamoeba hartmanni is just like histolytica, and but it's 5 to 12 micrometer, and they ingest the bacteria instead of RBCs. E. histolytica is bigger than Entamoeba hartmanni. It's a non-pathogenic. Entamoeba coli, it's irregular peripheral chromatid and concentric karyosomes, 15 to 20 micrometer. Karyocytoplasm with the many vacuoles and ingested bacteria and short blunt multiple pseudophore and sluggish non-directional motility. It's around 10 to 15 micrometer. It's cyst, and the nuclei occasionally chromatid bodies may splintered in may have glycogen vacuole. It's also non-pathogenic. Endolymix nana is no peripheral chromatid. It's large irregular karyosome, and it's also like the histolytica, and it's 12 to 2 to 12 micrometer in size, and single nucleus, finely granular vacuolated cytoplasm, blunt pseudophore, sluggish non-progressive motility, and the cyst of Endolymix nana is 5 to 10, round to oval, and usually nuclei, no chromatid bodies, may have poorly defined the glycogen masses. It's non-pathogenic, one of the smallest amoeba in intestinal amoebas. Iodoamoeba busheli have no peripheral chromatid large karyosome surrounded by the layer of the small granules, and it's like histolytica, and 8 to 20 micrometer, grossly granular cytoplasm with the vacuola and the bacteria, blunt pseudopods, sluggishly progressive motility 5 to 20 micrometer, ovoid, one nucleus, prominent glycogen vacuole and iodine cyst, non-pathogenic, only amoeba with just one nucleus in the cyst. The Iodoamoeba busheli and Endolymix nana have two distinguishing characteristic. One is the smallest amoeba, and Iodoamoeba busheli is the only amoeba that have only one nucleus in the cyst, and all of these four are quite resemblance with the Interamoeba histolytica, that is a pathogenic form. And if we observe that Interamoeba histolytica do not have ingested bacteria, and all of these organisms have a common characteristic that they ingest the bacteria instead of RBCs. Blastocystis pseudomonas BH is a normal flora of the gut, and it's belong to the Epicomplexa phylum, and can infect the main birds and reptile. It contains the large central vacuole commonly found in the GI of healthy individual, but in immunocompromised host they can cause infection. So it is opportunistic amoeba. Blastocystis pseudomonas is have a nucleus up to 4 pushed to the sides, and it's around 6 to 40 micrometer, large central body, as they don't have a cyst form, and it's a opportunistic, and should be quantified, formerly classified as a yeast, and now considered as an amoeba. The infection of Blastocystis pseudomonas is completely dependent upon the quantitations. If its quantity is moderate and numerous in a particular sample, that indicate the infection of Blastocystis pseudomonas, and also you have to correlate with the immunocompromised condition of the patient. Flagellates of intestinal and urogenital tract are Dientamoeba fragilis. Number one, it's in GI flagellates. They have trophozoite form but don't have a cyst form. Transmission is fecal oral or oral anal. It's a pathogenic organism, and if 5 to 15 micrometer, and 80% have two nuclei. No peripheral chromatid. Karyosome is mass of 4 to 8 granules. It's you can see inside the nucleus there is a karyosome, and karyosome have 2 to 8 granules, and this organism is a pathogenic organism. A flagella only visible with the electron microscope. It's very common organism in US. Giardia lambia is from the phylum Sarcomastigophora. It have trophozoite and cyst form. Trophozoite has a badminton racket shape, and trichrome staining and entero test is used for this organism to identify, and the cyst form is a monkey shape. In intestinal flagellates, there is one another organism that is very important. It's intestinal and urogenital tract flagellates, and that is Giardia lambia, is known as Giardia intestinalis and Giardia duodenalis. It's a pear shape, 10 to 20 micrometer, 2 nuclei with the large center karyosomes, 4 pair of flagella, interior and ventral sucking disc, 2 exostyla, 2 median bodies, old man in glasses, falling leaf motility. If we see the picture of the trophozoite form, and the upper head portion, it is a sucking disc on a ventral surface, and it have 4 flagella surrounded by the body and 2 nuclei, and median have parabasal body, and one line that crosses the body from head to tail is exoneme. In the cyst form, which is ovoid 8 to 19 micrometer thick double wall, cytoplasm shrink away from the cell wall, 2 to 4 nuclei at the interior end, and median bodies are exostyla nuclei. There are total 4 nuclei in this cyst form, two of the each side and cyst wall, and exonemes is still there, which divide the body into 2 parts. Transmission is ingestion of the cyst into contaminated water and oral anal pits, and it's a pathogenic common in the US. Entero test, a patient swallow the capsule attached to the string, and string withdraw after 4 hours. Mucus examine, and detection of the antigen can also be done by this mucus. In intestinal intestinal flagellates, there is 2 non-pathogenic flagellates that are Chilomestric misnelli and Trichomonas humanus. Both of these organisms are non-pathogenic, and they are from fecal-oral contamination or ingestion of the cyst. And Chilomestric misnelli, the broad interior tapping posterior and 3 flagella at interior, 1 at posterior, and spiral groove running length of the body, and prominently cytostome border by the fibrillaris, single nucleus at interior stuff, rotatory motility 6 to 10 micrometer, round with the bug at interior central and large nucleus, and lemon cyst, just like a lemon cyst. And the Trichomonas humanus is a prominent exostyla extending through the posterior and 4 interior flagella, undulating membrane entire length of the body along the costa, 1 oval nucleus with the small karyosome, and they don't have the cyst form ingested by fecal-oral contamination, and they are not pathogenic, easily to identify living organism rotatory motions. In flagellates of intestinal and urogenital tract are the most important in these are Trichomonas vaginalis, and this is the only flagellate in the urogenital tract, and diagnosis by the Whitmouth or jerky motility culture, rapid antigen test and PCR is also available. The transmission is through sexual, and this Trichomonas vaginalis don't have a cyst, only a trophozoite form, which is 5 to 18 micrometer, and 4 interior flagella, undulating membrane that extend half the body length. It's the nucleus are in the center, and exostyla, you can see it's divided the body into 2 parts. Costa and undulating membrane is 1/2 of the body, and 4 interior flagella, and posterior exostyla is also including in this uropflagellates. Among the intestinal ciliates, the Blatidium coli, it's ovid, usually 40 to 50 micrometer, but can reach to 200 micrometer, covered with the cilia, and funnel shape cytosomes, micro nucleus, and macronucleus vacuole, rapid rotatory motion, round cyst are round, 45 to 65 micrometer thick, refractile cells wall, no cilia is attached. Ingestion of the cyst is through the water, and it's a pathogenic organism, the only ciliate pathogenic to the human, and it's the largest protozoan of the human. It's common in the pig farming or living with the pigs. Blautidium coli, as we explained, having the two nuclei, one is macro and other is micro, and mainly cause the dysentery. It's from the phylum Ciliata, and it's the only ciliates that are infectious to humans. It have two form trophozoite and cyst form, and highly motile and usually immobilized with the help of the formalin, and a high rate of infection with the pig forming. Among the class of intestinal sporozoan, Isospora belli, it's a very important organism. Is the infection start from the ingestion of the oocysts in the fecal contaminated water and food, and diagnosis is also the direction of oocysts in the feces, which is 30 to 12 micrometer, transparent, 1 to 2 sporoblast. It's in the human is a definitive host, and cause anorexia, nausea, abdominal pain, diarrhea, and possible malabsorption. Another sporozoan are Cryptosporidium parvum. It's also ingestion of the oocytes from the food in the contaminated water and animal feces, anal, oral route, direct contact with the infected individual and animal. It's for the diagnosis, the modified acid fast stain of the feces red spherical bodies 3 to 6 micrometer and 4 sporozoites amino acid kits. Another the important opportunistic infection in AIDS patient cause the chronic diarrhea in the immunocompromised acute self-limiting diarrhea in immunocompetent patients. The another sporozoan in intestinal sporozoan are cyclospora. Kinetogenesis is through the contaminated food and water, and the oocytes in the direct wet mount is 8 to 10 micrometer, and visible staining with the modified acid fast and autofluorescence under the UV light, bright blue at 365 nanometer and mint green at 450 to 490 nanometer. It's a cause of a prolonged diarrhea, cyclospora. Microsporidia is ingestion of the spores and chromotroph of a cyclophore white stain of the formalin preserved stool. Spores are 0.5 to 4.0 micrometer. It's obligate intracellular parasite opportunistic pathogen, mainly in the AIDS patient, cause the prolonged diarrhea, cause disseminated, and molecular studies indicate it's related to the fungi. Isospora species are Coccidia, intracellularly phylum Epicomplexa, non-photosynthetic plastid. It's oocytes, and then it's converted into the sporozoite, merozoite, mesont, gametocyte, zygote formed during its complex life cycle. Gametocyte zygote formed during its complex life cycle. Oocytes infection caused by the ingestion of oocytes. It's elliptical and refractile, cause the diarrhea, vomiting, and death. It's the most common agent of a travel diarrhea. Fecal specimen have refractile oocytes resistant to spores, and we can also use the modified acid fast bacilli staining for the fecal specimen to confirm the oocytes of Isospora. The drug of choice is Septran. C-Par1 is also sporozoan, and that's belong to the Epicomplexa. It's oocytes and sporozoite, meront, merozoite, gametocyte, and then zygote. It's a fecal-oral route. It have a complex life cycle, and it cause the watery diarrhea, and not host specific. In fact, the human cattle dogs. Diagnostic technique are centrifugal flotation and modified acid fast staining, commonly used to detect the Cryptosporidium oocyte in the stool specimen. In intestinal nematode, trichoria is one of the important weep worm which cause the eosinophilia, and eggs require the developmental period in the warm soil before infective, and the route of infection are ingestion of infective eggs. The diagnostic stage are brill shaped ovum, which is 50 to 23 micrometer, and brown or yellow, clear plug at either ends. If we see the picture, the undeveloped unicellular embryo is inside, and the smooth shell surface, thick yellow brown shells color due to the bile staining, and hyaline plug at each pole is present. Trichoria is a weep worm, and the male worm is relatively smaller than female, and one fertilized female lay 1000 to 7000 egg per day. Approximately 10% of world population is infected. Infection takes place by ingestion of embryonated egg through the contaminated food and drink. Larvae hatch into the small intestine and develop into the adult larva in this adult worm line several years in cecum and ascending colon by brewing the mucosa, mostly asymptomatic, but heavy infection cause the abdominal discomfort, anemia, bloody diarrhea, rectum proliferates, and appendicitis. Another intestinal nematode are Enterobois vermicularis, and route of infection or ingestion of infected eggs through contaminated water and food. Its diagnosis stage is a wide ovum, which is 50 to 20 micrometer, one side flattened and colorless larva inside. It's a pinworm, common in the US, common in the children. Resistant egg in the home can infect other family member. Eaching one adult female migrate out of anus at night to deposit egg, and diagnosed by the pin worm pedals or cellophane tape prep. It's a thick wall, colorless shell. The developing larva is inside, which is colorless, and coiled larva developing into the egg, and one side shell is flattened. Enterobois vermicularis is a pinworm and nematode, and female is bigger than male, and adult worm lie into the buccal area where from the given female worm migrate to the night to the preanal to lay their eggs. Infection in the men by the ingestion of the egg or retro-auto infection also occur when the larva hatch at the perianal area, enter to the intestine and develop into the adult worm. This parasite infection is more common in the crowded and poor hygiene areas. Migration of gravid female worm to the anal area causes the itching and the sleeping disturbance, occasionally also cause the appendicitis, ulcerative colitis, vaginitis, and slapginitis in the female. Diagnosis is made by the detecting of the characteristic egg into cellophane tape preparation by touching the preanal region early in the morning. An intestinal nematode, the Ancylostoma, which is a hookworm, have 5 species, and among these 5 species, it's Ancylostoma duodenale and Nectar americanus is the pathogenic organism. It's a hook-like appearance of interior ends, and this organism is the Nectar americanus and the Ancylostoma duodenale are approximately similar to each other, but Ancylostoma duodenale are old world hookworm, and it is called hookworm because the hooks appearance of the interior ends. About 20% of our population are infected with Ancylostoma and this type of a hookworm, and these Nectar americanus and Ancylostoma duodenale are human intestinal hookworms, and other are animal hookworms. Adult worms are brown at the time of the passing. One fertilized female worm produce up to 1000 egg per day as long as for 9 years. The female worm are bigger than male, and Ancylostoma duodenale relatively bigger than and more pathogenic than Nectar americanus, and the eggs color are oval shape, colorless and oval shape, rubidiform larva, and then it is into the transfer into the filariform larva, non-feeding form. Infection takes place by the skin penetration by the filariform larva. Some also can infect through the oral route, migrate to the heart and the lungs, and then trachea and larynx and upper small intestine grow into the adult worms. Adult worms cause the anemia, heavy blood loss, hunger pain, pneumonitis, and the filariform larva cause the pruritus at the site of the skin penetration. Diagnosis is made by the eggs in the fecal specimen. Nectar americanus through the skin penetration or ingestion of the filariform larva. It's ovoid ovum 40 to 60 micrometer, colorless, containing the segmented embryo, 2 to 8 cells stages, surrounded by the clear outer zone, which is thin, smooth, colorless shell. 2 to 8 cell stages of the cleavage is you can see inside in this picture. And this American hookworm is a ground itch larva migration through the lungs. Eosinophilia is the major prognostic marker, and it can cause to the severe anemia. Ancylostoma duodenale is same like the nectar, but have also name as old world hookworm.